arm that we're going to focus on today. And then later, we'll reverse direction and talk about the endocytic pathway, which uses a totally different set of cellular machinery to go from the plasma membrane, how proteins are endocytosed or internalized from the plasma membrane to the inside of the cell. But today, again, we're going to focus on the secretory pathway, and we're going to focus on the very beginning of the secretory pathway, the rough endoplasm, what happens in the rough endoplasmic reticulum. And it's a nice segue as we talk about membrane proteins, because it is this beginning part of the secretory pathway that explains how proteins are synthesized and put into the membrane in the first place. So as we talk about protein synthesis, protein translation, let's recall that ribosomes accomplish protein synthesis. And recall that ribosomes, the protein synthesis translation occurs in the cytosol. It's accomplished by large protein machinery called ribosomes. And the ribosomes have a small unit and a subunit and a big subunit. And some ribosomes make cytosolic proteins. So what happens, as a review that you've had in many biology classes, perhaps genetics, is the message that it was exported from, the messenger RNA that was exported from the nucleus is in the cytosol, and then the 5' end of the message is a docking site for ribosomes, and the small unit and the big subunit get together on the 5' end, and then the ribosomes start synthesizing proteins. That's the green thing you start seeing emerging here. And a polyribosome is a single message with many ribosomes docked on it. You can see it here, and you can see that the synthesis begins at the 5' end, where there's a small protein. Here's and then goes to the 3' end. So this ribosome has been in here longer. Here's a larger protein, and these proteins would end up being cytosolic proteins. They'd be proteins like tubulin, actin. Think of all the cytosol as a super protein-rich environment. Metabolic proteins, all kinds of proteins are going to be cytosolic proteins because of this process. But what you may not realize, all of you have heard of the rough endoplasmic reticulum, and what you may not realize, it is this same pool of ribosomes that begin synthesis and dock onto the rough endoplasmic reticulum and synthesize proteins into the rough endoplasmic reticulum. So because of electron micrographs and other diagrams, many people think of the rough endoplasmic reticulum as having permanently docked ribosomes on them. But that's not, and permanently rough, that's not how it works, or a different set of ribosomes. It's the same ribosomes that are in the cytosol that synthesize both cytosolic proteins as well as proteins that are going to end up in the rough endoplasmic reticulum. In fact, the messenger RNA start out in the cytosol as well. And instead, what happens with a protein that's going to end up in the rough endoplasmic reticulum is that the small subunit and the large subunit dock onto the rough endoplasmic, dock onto the messenger RNA, and then something happens where that ribosome goes over to the rough endoplasmic reticulum and starts putting the protein into the rough endoplasmic reticulum rather than putting it into the cytosol. And that's the process we're going to look at today. The signals and machinery that are responsible for putting a protein into the rough endoplasmic reticulum rather than into the cytosol. And that machinery has nothing to do with the ribosomes. Those are just following directions. Rather, it's other cellular components that decide whether a protein is going to be put into the rough endoplasmic reticulum or into the cytosol. So what are these proteins that are going to end up in the rough endoplasmic reticulum? We've already talked about some of them. So these are going to be proteins that are going to be membrane proteins that are going to be on the plasma membrane, proteins like the LDL receptor or other receptors, proteins that are going to be in membranes that are along the secretory pathway, as well as proteins that are going to be secreted. So all of these proteins are not initially put into the cytosol, rather they're synthesized directly into the rough endoplasmic reticulum. So let's look at that process. And this recaps what I just said. So there are 3 ribosomes in the cytosol, same subunits as the membrane-bound ribosomes, the rough endoplasmic reticulum membrane ribosomes. The free ribosomes in the cytosol synthesize proteins into the cytosol, and those proteins might end up as cytosolic proteins, which aren't here, but would be actin tubulin and metabolic enzymes, all the proteins in the cytosol. Or there are also organelles where the protein, after it's synthesized in the cytosol, is imported into the nucleus, into the mitochondria, into chloroplasts, or into peroxisomes. And we'll have more to say about this importing process later in the course. But all of these proteins, again, are fully synthesized into the cytosol and later imported into these organelles. And again, we'll have more to say about those signals in a couple lectures. Today we're going to focus on this arm right here. These are proteins that are synthesized into the rough endoplasmic reticulum. And where they could end up is in secretory vesicles that are going to end up in the exported to the outside of the cell. They could end up in never secreted from the cell, but as lysosomal enzymes. They could end up on the plasma membrane if they're integral membrane proteins. And then not drawn here, these could also be proteins that are going to be in the Golgi or trans-Golgi network or anywhere along the secretory pathway. So all of these proteins are not synthesized in the cytosol. Rather, the ribosomes direct them into the rough endoplasmic reticulum, and then they end up where they're going to go. So what are the signals then that decide if a protein's going to end up in these organelles or somewhere else? And that's what we're going to talk about today. How do ribosomes end up on the endoplasmic reticulum? And let's think about what the endoplasmic reticulum is. And this movie shows a cell that's been labeled with it's a living cell that's been labeled with a GFP protein that ends up in the, it's been tagged in a way that it'll end up in the rough endoplasmic reticulum. So the cell it's a very flat cell. It looks like this right here. And you can, this is the nucleus, and it's excluded the rough endoplasmic reticulum, so it looks like it's blank. And what you can see is the rest of the cell sort of has this lacy network. And you can see that the rough endoplasmic reticulum in this living cell is a lacy network that extends from the nucleus all the way out to the plasma membrane. It's all over the cell. So cells are just full of rough endoplasmic reticulum. And this movie was made some years ago. It's one of the first GFP movies made, and it's made by someone who was a postdoc at NIH, but he was actually an undergrad here at UT, majoring in biology before he went on to do pretty famous work with GFP as a postdoc. Okay, so what information is it then that makes a protein go to the endoplasmic reticulum as it's being synthesized, as it's being translated? The message is directed to the rough endoplasmic reticulum rather than going to the cytoplasm. The story of the sort of machinery, the cellular machinery that dictates whether a protein is synthesized into the rough endoplasmic reticulum or is synthesized into the cytosol, and whether it's where it ends up, was first worked out by Gunter Blobel's lab at the Rockefeller. And he ended up winning a Nobel Prize along with some other people for the body of this work, which was a lifetime of work. And the approach they took was biochemical, and one of the first things they did was to compare the sequence of proteins that ended up being imported into the nucleus, exported from the nucleus, imported into mitochondria, imported into plastids or chloroplasts, peroxisomes, ER, or returning to ER. And we're going to talk about these signals as we go through the class. So the signal sequence, the ER signal sequence, was the first of these sequences worked out. And what these signals are they can be thought of as zip codes, sort of like on a letter, where a zip code decides where a letter is going to go. These sequences on proteins decide where a protein is going to go. If a protein has this sequence, it's going to end up being imported, synthesized in the cytosol and imported into the nucleus. If it has this sequence, it's going to end up, once it's in the nucleus, exported from the nucleus, etc. So there are signals on proteins that dictate where a protein is going to go. And if we think of ingredients, can everybody see this board okay? The first thing we're going to talk about is, so we talk about who, what, where. The first one, which was just called the signal sequence. How many people have heard of signal sequences before? Probably in a previous class. We're going to modify that now that it turns out there's lots of signals to call it the ER signal sequence. So the ER signal sequence, so we can distinguish it from the other one, is on, what it does is it directs a protein to the ER, and it's on the, where it is, it's on the N-terminus of the protein that's being synthesized. So here's an example of an ER signal sequence right here. You can see that this is the amino terminus right here, and this is the protein could continue right here. And this is the ER signal sequence. Those of you who can see the amino acid sequence, what can you say about this sequence, the ER signal sequence right here? It's very hydrophobic, isn't it? So these are generally hydrophobic amino acids. So it's a stretch of hydrophobic amino, it's a loose sequence. We'll see that some of these sequences have to be the exact amino acids. The ER signal sequence is pretty loose. So it's a bunch of amino acids, 10 to 15 or so, that are close to the amino terminus. So let's look more closely at that. The ER signal sequence, here's the one for growth hormone. This is a protein that is synthesized into the rough endoplasmic reticulum. So it's the targeting signal to go to the ER right here. And we can see right here, here's the initiation methionine. Here's 10 or 15 or so hydrophobic amino acids. You can see there's a little charged one right here. That's important. Toward the positive charges tend to be a charged one right here. Here's another example, serum albumin. That's a protein that's going to be synthesized, secreted from the cell. You can see that it doesn't look anything like this protein. ER signal sequence. It's hydrophobic. Here's a positively charged amino acid. It's closer to the amino terminus. Here's another one for HIV glycoprotein. It's a bunch of hydrophobic amino acids. Again, what these amino acids have in common is they're hydrophobic, and they tend to be preceded by some positive charges, something like arginine or lysine. So when you compare all the ER signal sequences from proteins that are going to be secreted, what you find is that you find an amino terminal stretch of nonpolar hydrophobic. Okay, we use the word residues a lot. It's something cell biologists and biologists do, so I'll slip into that terminology. A residue is an amino acid. So a residue is an amino acid. Same thing.